and while you get that up, okay, great. So our final speaker is Ali Phillimore, who's a Chancellor's Fellow in the School of Biological Sciences at University of Edinburgh. Ali is a leading evolutionary ecologist who studies impacts of global change on species interactions and fitness. And he's specifically interested in how the timing of these key life history events can have major effects on fitness. And given that, you know, all of the projections and the evidence show that phenology will advance, you know, this kind of work I think is crucial to understand what the consequences of that will be for most species. So it's a great pleasure to welcome Ali and to invite him to give his talk, Timing It Right in a Changing Climate. Thank you.
Thanks very much for the introduction, and thanks for staying around, everyone. So what I'm going to be talking about today then is phenology. And I'm sure you all know a bit about phenology, the timing of recurrent life history events. This can be the flowering times of plants, flight times of butterflies, breeding times of birds or frogs, and many of these different kind of spring events. And phenology seems kind of a really interesting area in terms of capturing the imagination of amateur naturalists and citizen scientists, and also increasingly capturing the imagination of evolutionary biologists and ecologists. And I want to explore a little bit why evolutionary biologists and ecologists have become so interested in phenology. So as we'll see in a few slides of my talk, and as you all know, spring phenology is very strongly affected by spring temperatures, warmer temperatures and earlier timings at high latitudes. But phenology also is very important in determining the abiotic conditions an organism is exposed to. So imagine a frog that spawns too early and might have its spawn exposed to late frosts. And it's also very important in determining the interactions that are available with the same and with other species. So opportunities for mating or interactions with competitors or availability of resources. So because of phenology actually mattering for abiotic conditions and biotic interactions, then it's expected to have a really pronounced impact on reproduction and survival, or in kind of Darwinian terms, the struggle for existence. And so for an ecologist and evolutionary biologist, because spring temperatures are expected to affect phenology, and phenology is expected to matter for fitness, then this is a particularly interesting place for us to look for impacts. Now I'd like to give a kind of little bit of a historical context to phenology, where we can see where Darwin fits into it. So if we go right back to 1736, and Robert Marsham, very local to Norwich, so he's on the Stratton Strawless estate just 10 miles north of Norwich, he started collecting observations of 27 different indications of spring, so things like the first swallow, the first oak in leaf, and so on. And he continued collecting these observations throughout his life. And towards the end of his life, he reported them to the Royal Society. And then the miraculous thing is his descendants continued collecting these records right the way through until the 1940s. And then a bit later, from about the 1880s through till the 1940s, the Royal Meteorological Society produced an annual phenological report where people submitted phenological observations from across the UK. So this must have been a very early case of citizen science ecology. And you can see these beautiful kind of isocare maps of equal phenology across the UK. And then unfortunately in the 1940s, really it was people were persuaded that phenology was just the preserve of the amateur naturalist. So they stopped recording. And we can see where Darwin fits in this. He actually doesn't say an awful lot about phenology. I found a quote from his Voyage of the Beagle, where he's in Patagonia and he says, on the 15th of September, a few animals began to appear. And by the 18th, everything announced the commencement of spring. The plains were ornamented by the flowers of a pink wood sorrel, wild peas, inothera, and geraniums, and the birds began to lay their eggs. And what follows in this this quote, he then goes on to explain that it's all driven by the rising temperatures that were in the preceding days. So Darwin had a very clear understanding of what was driving phenology. So let's bring kind of phenology research forward. Here we have the famous climate stripes. And from about the 1940s, the British Trust for Ornithology were collating people's observations of nest records. So this is where really avid ornithologists go and either find nests or use nest boxes and submit observations of when birds were first laying their eggs. And then more recently, there's been other citizen scientists, such as the very successful Nature's Calendar in the UK, which started in 1998, and they now have many millions of observations of the first spawning dates of frogs, the first leafing dates of oaks, first swallow, and so on. So I'll be coming back to the value of some of these citizen science data later in the talk. One of the nice things actually with Nature's Calendar is they have a really good website that allows you to visualize how phenology is changing through the UK by sliding this bar at the bottom. So you can see the gradual spread of frog spawning through the UK, for instance. So some of my own group's work looking at the effects of spring temperatures on phenology from these citizen science data sets shows really quite pronounced shifts. So for the first leafing dates of many tree species in the UK, with Nature's Calendar data, we find there's around about a 1 week advance if the year is 1 degree warmer. For the flowering times of plants such as cuckoo flower, garlic, mustard, we find about a 6 days advance per degree. For butterfly flight times, for the univoltine species, we find around about 6 days advance per degree. And for birds, these are for mainly whole nesting birds with BTO data, we find around about 4 days advance per degree. So the main question I'm kind of exploring in this talk today is, are populations able to get timings right in a changing climate? And there's 3 parts of the question I'm going to explore. The first is, how much phenological plasticity is there? By which I mean, how much are individuals able to just adjust their timing from year to year? The second one we'll look at is, how much is the optimum timing shifting? And by kind of putting the first 2 parts together, we can ask, well, how much shortfall is there, and how much will populations need to evolve to keep up, and are they able to? And we'll see there's been quite a kind of different amounts of progress on these different questions. So Darwin also had something to say on phenotypic plasticity. He says, when variation is of the slightest use to any being, we cannot tell how much to attribute to the cumulative action of natural selection and how much to the definite action of the conditions of life. So this is from the origin. And he goes on to talk about the fact that as you go to colder environments, you get thicker and thicker fur. Now, of course, we now think of plasticity as itself is very potentially adaptive, and that's something I'll talk about in relation to phenology. So how much phenological plasticity is there? Well, I'm going to show you a few slides based on a very quick analysis of data we've collected ourselves. We've got this transect through Scotland, where we monitor phenology of trees, caterpillars, and birds at 44 sites. And what I've done here is I've taken a single oak tree and its first leafing dates in different years. And we can see that the difference between the earliest year and the latest year is over 2 weeks. So this is an individual having a pronounced variation in when it comes into leaf. And then we can plot the timing of that single oak tree's leafing in relation to the March-April average temperature at that site in each year. And you can see, although I haven't fitted a regression line to these very few data points, you can see a pronounced negative trend. And I think the slope of that's around about a 7 days advance per degree. So really pronounced plasticity from the oak trees. And we can also do similar types of analysis for the blue tits. So we can go to individual blue tits that we've ringed on the nest and find the few lucky individuals that survive more than 1 or 2 seasons and see how their timing varies. So this is one individual female, Z632398, and the timing of its egg laying in different years. And what I've done in the next slide is I've looked at all the individuals that survived for more than 3 years, how much the temperature that they experienced deviated in each year, how much their egg laying deviated in each year. And from that, we can estimate an overall kind of average plasticity of these birds. And you can see that individual birds lay their eggs earlier in a warmer year. So this was a very quick analysis. From White and Woods, people like Ben Sheldon have much better analyses that show that great tits have around about 4 days per degree plasticity. So the conclusion of this first part of the talk is that when we look for evidence of phenological plasticity, we actually find quite a lot of it. So the next question is, how much is the optimum timing shifting? And this proves to be a much harder nut to crack. Now, if we want to identify how the optimum is shifting, we first need to estimate the optimum timing in an individual year. So we need observations of many individuals with different timings, and we need to quantify their fitness. And if we do that, then we can fit a curve through it, and we can estimate when the optimum timing is. That's just for one single year. And if we want to ask how the optimum timing is shifting, we need to do that for many different years, and then estimate how those optimum timings are shifting, either in relation to year or in relation to temperature. So you can see that the data required for this requires very long-term studies, but also a great amount of detail to them as well. And as a consequence, we've had very few estimates of this. I'm just going to give you an example of the two that I know of. So this is a study of great tits studied in the Netherlands from 1973 to 2013, although their study is ongoing. So this is the famous Hogevaloo population. And they applied an approach very much like what I described to these hole nesting birds. And they found that the optimum laying date advanced by about 5 days for every 1 degree warmer the spring was. So quite similar to what we've seen estimates of plasticity are for species like this. And here's another study of dippers on rivers in southern Norway. Again, a long-term study from 1979 to 2013. And they found that the optimum lay date was advancing by 1 day per year. So from these two studies, we've got some evidence that the optimum timing is getting earlier and is advancing with warming. But we're really a long way from getting these types of estimates for many species. It's just too much work involved to do these long-term studies. And so this got myself and colleagues thinking about whether there's other ways we could try and make similar inferences. Now, we know temperature varies a lot from year to year, but it also varies a lot geographically. So in the UK, much warmer in the south than in the north. And so what we've been exploring is whether you can do a kind of space for time substitution, but where you use the spatial phenotypic response to temperature to inform us about kind of long-term or equilibrium responses. So potentially even optimum responses. So I'll just take you through a method that I developed along with Jared Hadfield and Owen Jones, where we can try and detect local adaptation from citizen science data. And as I've said, there's this abundantly available citizen science data collected for many locations and many years. So imagine that, imagine we've looked at a single population in different years. We know what the temperature Q is. We know what the phenology is, and we can do a regression to see how does phenology change as temperature changes. And so that should be due to plasticity and potentially a contribution of adaptation. But if we're looking at kind of a fluctuating temperature change and a short time period, then perhaps adaptation's a very small contribution. Now imagine we're looking at a relationship between temperature and phenology over many different sites, many different populations. So that relationship should be due to plasticity plus the contribution of local adaptation. And so this is where the kind of equilibrium assumption about the spatial relationship comes in. Now, if we assume the plasticity is a constant, the difference between those two slopes gives us an estimate of local adaptation. So our first test case for this method was frog spawning based on tens of thousands of observation of first spawning dates in the UK. And we, in this first model, we found that January temperature was the best predictor of spawning date. So each one of these lines here on the left shows the average slope across time, across different years, for different populations that we've subdivided in the UK. So we find that the frogs spawn around about 3 days earlier for every 1 degree the year's warmer. If instead we look at the average spawning date and the average temperature in different grid cells, we get this spatial relationship, which is much, much steeper. So about minus 13 days per degree C. And so from that, we made the inference that the plasticity was minus 3 days per degree C, and the local adaptation, the difference between these two slopes, was significant. Now we can take these slopes and multiply it with predicted changes in UK temperature. So this was from an old projection of UK temperature increase for 2050, projecting 2.8 degrees in the southeast and about 1.6 degrees in north of Scotland. So we can multiply this temperature increase by the plastic slope to see how much we think the frogs should advance via plasticity. And we can also multiply this spatial slope to see how much they need to advance to stay as adapted as they currently are. So how much do they need to adapt to stay equilibrium? And we can see that's a much more, much larger advance they need. So they need about 37 days in the south of England. So there's a huge kind of shortfall between how much they'll shift their timing via plasticity and how much they might need to shift. So we've taken this method, we developed a little bit further, and we've applied it to the egg-laying dates of these 4 bird species, so blue tit, great tit, chaffinch, and pied flycatcher. You can see very pronounced gradients across the UK and when they lay their eggs, both latitudinal and longitudinal. And you can see that all 4 species have advanced their egg-laying date over the time period we were looking at. So I think for great tits, they would advance by around about somewhere between 10 days and 2 weeks. So I won't go into the underlying kind of methods and assumptions for this, but using a method that Jared developed, Jared Hadfield, we were able to estimate how much the optimum timing was shifting for these birds. And we were finding that for every 1 degree warmer, all 4 of these bird species were shifting their optimum egg laying date should shift by about 4 days. So the question is, how well can plasticity track that? Well, here is our estimates of plasticity for the same species. What we can see is the estimates of plasticity are remarkably similar to the estimates of the optimum, the shift in the optimum. So we inferred that plasticity and laying date was very similar to the optimum slope, and that means that plasticity is itself very adaptive. And there's one exception that I'll come back to, and that's the pied flycatcher. Here you can see the optimum is shifting by about 4 days per degree, but the plasticity looks a bit shallower. So this brings us to the final question I had then is, will populations need to evolve to keep up, and are they able to? So we'll look at kind of how much is the optimum shifting and how steep is the plastic response, and can it track the optimum? So here we have a relationship between temperature and phenology, the optimum relationship. And you might find a bit faint in blue below, that's the plastic response. And that would be a case where plasticity tracks the optimum timing. And I'd say that some of the species we're looking at, things like the blue tit, the great tit, and the chaffinch, seem to really fit with that scenario. Another scenario is where the optimum relationship is steeper than the plastic response. And as we get to warmer and warmer temperatures, there's a kind of a lag that's going to need to be made up. So these are the situations where the populations might be kind of lagging behind climate change and where adaptation might be required. And so a case where that may exist would be the pied flycatcher that I already mentioned. Well, a recent review, well, actually it's no longer recent, it's 2012, but I think the statement still stands. This review said that the genetic evidence for climate change-driven evolution is still scarce. And there's a variety of reasons that I could perhaps come to later of why the evidence for climate change-driven evolution might be scarce. But we do have some really nice case studies. So this is the purple pitcher plant mosquito. And in the United States, this has been shown to shift the phenology of its diapause. That's probably due to climate change. And also Van Asch et al. Showed that the winter moth caterpillars shift in their phenology and has been a kind of an evolutionary response. So both of these have done what are known as common garden experiments over time, where the environmental conditions are controlled for. Okay, so that brings me back to the pied flycatchers. So unlike the other bird species I talked about, which are all resident breeders in the UK, the pied flycatcher has to migrates to West Africa and then decides to kind of fly back to Europe in March and April, which means that while it's traveling back from West Africa, it's not able to pick up on local temperatures and local conditions on its breeding grounds. So that might be part of why it has kind of shallower plasticity than the resident bird species. So what this really elegant experiment from Barbara Helm and colleagues did was they took individual nestlings out of the nest, hand reared them over the course of a year, and then looked at their spring and the autumn phenology in captivity, but in a very controlled environment with a fixed temperature. So again, a common garden experiment. But this is a common garden experiment through time again. So they did this first in 1981, and then much later in 2002. And over this 20-year period, you can see there's a shift in the timing of male development of around about 2 weeks advance. So although these are very small sample sizes they had, but I think this reflects what a painstaking, challenging experiment it must have been. But I think this is the best evidence we have for a shift in phenology of any vertebrate species. Sorry, an evolutionary response of any vertebrate species. Okay, so coming back to the questions I set out earlier, are populations able to get timings right in a changing climate? Well, in terms of plasticity, we find substantial adaptive phenological plasticity in response to temperature. So there's really loads of evidence for this. There's some accumulating evidence that the optimum spring timing is getting early with warming. I think it's quite intuitive that it should be getting somewhat earlier, but what we really lack are precise estimates, and we don't really have a diversity of precise estimates for a diversity of species. It's really mostly focused on birds. And where plasticity is not enough to track the shift in the optimum, then populations may be evolving earlier spring phenology. But again, we don't have a lot of evidence for this yet. So we don't know how fast, how able to evolve different populations are going to be. And just before I close, I just want to kind of come back to thinking about Darwin's tangled bank that he described so beautifully. And all of the analyses I've been talking about today treat populations as though they are they can be considered in isolation. So how does this species respond just to temperature? And yet, as I said at the very beginning of the talk, one of the really important roles of phenology is scheduling interactions with other species. And we can see this if we think about this tangled bank. So we can see the competition for light between these species on the herb species on the forest floor. So whether it's wooded enemies trying to get a head start ahead of bluebells, this competition for light. Then we see a competition for light for all of them to try and to develop before the canopy completely covers them and shades out the light. And then amongst the tree species, we see competition between early leafing species like birch and late leafing species like oak. And all of these tree species then have interactions with caterpillars that are feeding on them, birds feeding on the caterpillars. And so there's lots of potential that if different species are shifting their timings at different rates, that this could have effects that ripple through these whole communities. And I think the best, sorry, again, this is a very blurry slide, but the best illustration we have that this could be problematic comes from this lovely meta-analysis by Steve Thackeray and colleagues using citizen science phenology data, where they look at these 3 different trophic levels in the UK: primary producers, primary consumers, and secondary consumers. And you can see that the primary producers and the primary consumers are shifting their timings much faster than the secondary consumers, leading to this idea that there might be kind of increasing mismatch. And what we don't know is whether these mismatches have major consequences, or is there any sort of buffering? And I'm not able to answer that in any kind of robust way yet. And finally, thank you all very much for listening. And I really ought to say a big thanks to all the volunteer recorders, to all of these citizen science schemes, and thanks to collaborators and funders.